Next up, we'll have Dr. Kirsten Lyke from University of Maryland.
Great, thank you. So Adam, Stephen, thank you for the invitation. This has been a great meeting. Adam asked that I see, okay. Adam asked that I focus on some of the immunologic lessons that we're learning from our dengue human infection models, and I have to say I'm a clinician from the malaria world, so that strikes a bit of fear in my heart to try to talk about dengue immunology to this room, but we'll take it from there. I'm going to focus on a very narrow window, and that is the earliest weeks of the immune response. This is a collaboration. It's a Dengue Challenge Consortium, and we're trying to develop dengue human infection models to all 4 strains, collaborating with Walter Reed and SUNY Upstate, and we're funded by MTech. And this conference has really set up some of the challenges with dengue studies. So there's unpredictable patterns of dengue virus circulation. There are differences in the immune response to the serotypes, depending on the vaccine platform. We have few therapeutics. There's holes in our understanding of the immunopathogenesis. So we view the challenge as a discrete tool that can be used to ask specific questions. But for the purposes of this talk, we're really going to talk about allowing for the examination of the immunologic features of infection at the earliest moments and having a baseline. And so this is where we're at. This has been discussed in previous years. We have 3 strains or 3 serotypes optimized: dengue 1, 3, and 4. These are all Southeast Asian attenuated vaccine strains that were too reactogenic for vaccine development. So it's been repurposed as dengue human challenge models. They're all lyophilized, vialed, CGMP manufactured. We're going into dengue 2 challenge probably later this autumn, hopefully. And so essentially what we do is we're trying to optimize. We start with essentially 3 different dose levels, and we're trying to achieve some outcomes. So we want to have 90% development of RNA that lasts for more than 4 days, and we want mild and moderate symptoms. So importantly, we're aiming to have mild to moderate dengue-like symptoms. We challenge people by inoculation, and then we follow them depending on the serotype, but for dengue 4, which I'm going to focus on today, from days 2 to 16. By day 28, they're completely symptom-free, and then we have some additional immunologic time points that we follow out to day 180. And so just jumping to our results, so we challenged over a couple of different cohorts, 10 different individuals, all of whom developed detectable RNA as early as day 4, peaking at about day 7, and then lasting for approximately 9 days. And one of the participants had a little bit different kinetic profile for that viremia. But essentially, we obtained our outcome measures with the very first low-dose 95 PFU challenge dose. So we had extra funds, and we repurposed and redesigned the trial to build in a homologous rechallenge. 1 year later, we took 6 of those primary challenged individuals with 4 naive controls, and we backed up the detection day to day 2 and found that half of them developed, of the naive controls developed dengue 4 by day 2, but had very similar kinetics as to the previous cohort. So for the purposes of this talk, we have 14 primary individuals, and we have 6 homologous rechallenged individuals. And there's some kinetic variations depending on the strain. So we've noticed that dengue 3 and 4 tend to have an earlier onset, preceding dengue 1 by about 4 days, and dengue 1 peaks about 5.5 days later. And there's some differences in the peak viremia. For instance, dengue 4 peaks at about 10 to the 6th, whereas dengue 3 and 1 are a little bit higher, 10 to the 7th, even up to 10 to the 8th. And we've noticed that by and large, everyone has symptoms. Everyone has fatigue, headache, muscle aches. But there are some subtle variations. For instance, with dengue 4, we noticed that 100% of people have rash, and predominantly a total body rash, whereas in dengue 1, they do have rash, 65% perhaps, and not total body. So there's some subtle differences. There's much more fever in dengue 1, much less fever in dengue 4. That may be related to the viremia levels. But getting into the immunology, I have to shout out Christiana Cairo for the spectral flow cytometry, Joel Chua and Sigrid Camacho for a lot of the work that I'm going to describe here. And we're going to focus on the time points pre-challenge, day 4, which is what we had estimated as the onset of viremia, day 14, really the end of viremia, day 28, 90, and 180. And what we know about the earliest time points is that there's this huge viremia surge accompanied by NS1 detection. Reservoirs are the dendritic cells and the monocytes as well as B cells now. There's differentiation into macrophages, which through B cell activating factors and a proliferation ligand stimulate and activate B cells, which then result in this plasmablast surge and antibody formation. So as that template, we kind of set the stage. So first, our NS1 detection, we see that lag behind the RNA detection by about 2 days, but is on par with our plaque assay viremia results. We then turn to look at some of the myeloid response. So with monocytes, we gated out T cells, B cells, NK, basophils, dendritic cells, and gated on the CD88 positive cells. And just as a primer, the monocytes can really be divided into essentially 3 compartments. So there's the classical monocyte, which is canonical phagocytic monocyte, which can differentiate into dendritic cells or macrophages. There's this intermediate monocyte population, which is discriminated by both CD16 and CD14 expression. And this is more of an inflammatory subset. We used to think that this was a transitional subset, but single-cell RNA-seq really delineates this as a unique subset with its own fingerprint and its own unique functions. And then finally, the non-classical monocytes, which are characterized by high CD16, and these are a patrolling monocyte. So what do we see? So at the bottom is our classical monocytes, and this is a bit reflective of what we see in the monocytes as a whole, that there's a modest rise followed by a fall, and then a rebound effect. With the primary challenge, which you see on the left. When we're looking at the intermediate monocyte, it's much more interesting. So we see at day 4 that there's this increase in intermediate monocytes, followed by a decrease. In 2 individuals that had very high viremias, though, they continued to have evidence of intermediate monocytes even at day 14, and then there's a rebound that occurs all the way out to day 180. And then with non-classical monocytes, we again see this surge at day 4, and then a crash at day 14. We can't find them anymore. So we're presuming that they're trafficking, perhaps in the lymph node tissue. At re-challenge, however, we see no bump in any of the subsets. And just to reiterate, and doing some statistical analysis, both the intermediate and the non-classical really have this robust expansion. We don't have a day 2. That would be very interesting to add to see if we're even missing the peak. Now, we know that Fc gamma receptors mediate dengue virus entry into the leukocytes, so we wanted to examine CD64, CD32, and CD16A. We did not see any changes with the time points tested in regards to CD32 or CD16. However, with CD64, we did see in all compartments a pretty big, robust expression increase by day 14, and then a self-correction out to day 180. And importantly, nothing was noted during the re-challenge portion. Just to look at some unsupervised analysis, so in the middle upward panel, you can see that this is UMAP testing. The non-classical monocytes are in green, the intermediate monocytes in red, and classical in blue. And down below are a sequence of events across our time span. And you can see that, or maybe you can appreciate that at day 4, there's an increase in intermediate monocytes, and by day 14, really no detectable non-classical monocytes, which kind of recapitulate what we found in our spectroflow cytometry results. Now, looking at dendritic cells, this is really an active field. There's been a lot of movement. And so I put this cartoon from Bordelais et al. That really, through a successive number of publications, it seems that there is kind of this unique population now of dendritic cells called DC3s. So we know that the conventional DC1s and DC2s arise from a common progenitor, and there is a unique progenitor for monocytes. But there's this population of cells, and it's illustrated in single-cell RNA-seq, where dendritic cells are able to express CD14. And it really seems to be a bridging population between conventional DC2s and the monocyte population. You cannot stimulate the monocytes to differentiate into DC3s, and in the presence of an inflammatory background, you can move the expression of CD14 such that an inflammatory background has very high expression of CD14. We call these inflammatory DC3s. And so for the purposes of gating, we are gating on CD88 negative cells, CD1C positive, and then down below you can see we're using CD163 to kind of discriminate between our DC2s and 3s, and CD14 to kind of discriminate between DC3s and inflammatory DC3s, even though that's more of a continuum. And what we see is that there's perturbation of the conventional DC2s, where there's this decline that occurs over time at day 14, and then a rebound effect. And again, no response whatsoever in the challenge, and the homologous re-challenge. And then for the first time, we see DC3s in dengue, so it has a very similar pattern with this drop that occurs at day 14, presumably it's trafficking, and then a rebound effect. And we see inflammatory DC3s, which again, rise at day 4 and then drop off by day 14 with really no effect seen during the homologous re-challenge. So quite interesting. We don't have a day 2. That would be an important time point that might add more to this. And looking at Fc gamma receptor 1 again, CD64, we see upregulation in all 3 DC dendritic cell compartments that we analyzed. Plasmablasts, we were defining this by CD38, CD319, and we see a really robust explosion of plasmablasts that occur by day 14. We were thinking that we might have missed the peak at day 8, so it was interesting to hear Leah's data that there doesn't seem to be a correlation with peak viremia and the plasmablast development, and in our population, up to 35% of B cells down at the lower left panel are plasmablasts. No discernible plasmablasts with the homologous re-challenge. And then looking at unsupervised analysis, this kind of recapitulates what I just stated, where the plasmablasts in red at that lower right panel, if you look longitudinally at the bottom, you can see that by day 14, there's a really light up in the unsupervised analysis of our plasmablast expression, and really nothing to be seen in the homologous re-challenge. Micronewts tend to peak between day 15 and 28, and we did not see a bump in the subsequent re-challenge. In terms of subtype analysis, we see an explosion of IgM, we see IgG, and this was the only little tidbit we noted with the re-challenge. There did seem to be a little bump of IgG in our re-challenge population, and then we see IgA in the first exposure, but no discernible uptick at the second exposure. And in terms of plasmablasts, we see this large IgM plasmablast population, very little IgG, and we see IgA, it kind of drops off and then rises again. Looking at longitudinal differential gene expression, so this is some of Adam's work. And we see as we march out every 2 days, you can see that there's this explosion of upticks in gene expression starting at day 4, out to day 14, and then by day 28, it's completely gone. And these largely map to antiviral defense and inflammation. But there was a nice paper by Celine Hardy looking at some of the differences that occur between dengue 3, dengue 1, dengue 4. Dengue 4 squarely sits within the genes that light up for dengue 3, and dengue 3 has an abundance of more genes that light up. So there's interesting intersections to study there. This is a little bit more results on, so the inflammatory index is essentially pulling out 8 genes of inflammation. This has been validated by Adam in the SARS-CoV-2 model. It correlates with disease severity, and we can see with dengue and repurposing it for dengue that there's this correlation of the inflammatory index and peak viremia. But if you look at that purple line, that is our individual that had sort of aberrant viremia and a much more delayed detection of viremia. And this person is really unique. We didn't have any detectable plasmablasts. There's no detectable inflammatory index. If you look at them in principal component analysis, they're on an island of their own. And if you look at differential gene expression between that individual and all the others in the challenge model, there's this explosion of upregulated genes that are unique to that individual, and they largely map to metabolic function, B and T cell receptor upregulation, but also worryingly some renal and thyroid cancer markers. This individual did have a very high B cell percentage, which was persistent over the 2 years that we followed him, but really no evidence of any other abnormality. So it's a little bit of a puzzle. So just in conclusion, we see this transient expansion of intermediate and non-classical monocytes. We see a transient decline of classical and non-classical monocytes after the primary challenge by day 14, this decline of inflammatory DC3s, and this activated dendritic cell subset after the primary challenge, and upregulation of CD4, both in monocytes and dendritic cell subsets. Expansion of plasmablasts, expansion of mononuclear micronute titers and immunoglobulin subclass, and no measurable immune perturbation at the homologous rechallenge. One outlier participant had an aberrant curve, no plasmablast, no inflammatory index, so quite unusual. And these results we feel is fairly consistent with that's been reported for natural dengue infection, and allows us to really drill down deep and start to understand some of the things that are going on immunologically in the earliest moments of dengue infection. And so with that, I'll just thank all the individuals that have helped in the dengue consortium and turn it over.
Thank you so much.